some serious new challenges, as I mentioned. Tonight, we continue with our advocacy for the river corridor by calling for a common sense bridge restoration at Skinner's Falls, Milanville, and by advocating for water protection relative to the Delaware River Basin. I know from the faces on my screen that we can meet our challenges, so let's get to work. The next agenda item is item number 4, a virtual presentation, DRBC Microplastics Study by Jacob Bransky, Aquatic Biologist from the Delaware River Basin Commission. You have the floor, Jake.
Thanks a lot. We'll share my screen here, and hopefully we won't hit any technical issues. Screen. You guys seeing my screen now? Is it the note screen or the full screen?
Full screen.
Awesome. Alrighty, well, thanks Andy for the introduction. And yes, I'm Jacob Bransky. I'm an aquatic biologist with the DRBC. I will be talking today about topic of microplastics and also giving an update on some microplastics work that the DRBC has done over the past couple of years. Before I forget, I did get a text from Kristen just a moment ago. Her power went out right as she was logging on. And so she is making an attempt to get back on as soon as possible. Okay, so most of you are familiar with the DRBC, but for those that aren't, I'll do a brief bit of background. DRBC is a federal interstate agency comprised of the 4 states of Delaware, New Jersey, Pennsylvania, New York, and the federal government. Our broad responsibilities include water quality, water supply, drought management, flood loss reduction, watershed planning, regulatory review, outreach and education, and recreation. So ultimately, DRBC is responsible for water quantity, providing adequate, sustainable, resilient flow, and water quality, providing clean and healthy drinking water to the basin. I work on the water quality side of things. So in my department, we study lots of topics from fish and macroinvertebrates to dissolved oxygen and contaminants of emerging concern. And one topic that we've begun studying recently is microplastics, and that's what I'm going to be talking about here today. So let's start basic. What are microplastics? Microplastics are defined as small plastic particles, less than 5 millimeters in size, so roughly about the size of a sesame seed and smaller. They can come from a lot of different sources. They can come from microfibers from synthetic textiles. So like a lot of fleece material is made out of plastic fibers, which can then break down, get into the water cycle. They can also be found in microbeads in things like personal care products like face washes and toothpastes. A lot of plastics also come from just breakdown of larger items like tires or plastic bags, bottles, fishing line, styrofoam cups, things of that nature that get out into the environment and then break down over time. At the right here, there's some pictures of some particles that we found in our study, things like little pieces of plastic bag and little colorful fragments of a blue bead that we found, lots of fibers, which are tough to see kind of in the third one in the top row and the first one in the bottom row, just lots of fibers in the study. But those are just some examples of plastics that are out there floating around in the river. What are the impacts of microplastics on aquatic life? So this is a field that is still very much in development and being studied, but it is known that many species do confuse plastic particles for food items and they will ingest them. And this consumption of plastic by aquatic life can do several different things. It can fill their stomachs so that they reduce their feeding on other more nutritious food items. It can therefore reduce their growth and their reproductive ability. There's also some new research that shows that plastics can increase the transfer of toxic chemicals into aquatic species. So very, very, very small plastic particles known as nanoplastics can actually travel across cell membranes of animals and get into cells. And so if these nanoplastics are picking up toxins from the environment, they can then transport those toxins into the cells of organisms. So how do we study microplastics? Microplastics are a relatively new contaminant of concern, and there's a lot of different ways to look at them in the environment. We used 3 methods in our study. We used a net sampler, a simple grab sampler, and then a Niskin sampler. I'm going to talk about the net sampler first. So that's what you see at the top right here. This is basically a plankton net. It's the same type of net you would use to sample plankton in the water column. And we have fixed floats to the opening of this net so that the opening floated right on the surface of the water. So basically skimming that surface of the water. And then the net is tied to a rope to an anchor and oriented in the flow so that the flow of the water is entering the mouth of the net. And that flow just continues to keep that mouth open and collect particles for as long as you leave it out there. You could leave it out for a whole day if you wanted to, as long as there's not enough, debris to get the whole thing all clogged up and back the system up. When you're done sampling, you take the net out, you rinse everything down into the sample collection jar that you can see at the back end of the net there. And then you take it to the lab to process the samples. So this sampling is great for doing processing large volumes of water. You can filter thousands of liters of water through there over the course of a day, letting that sit out in the river. A downside to the sampling method is that it is a mesh net. So there are a certain portion of your particles could pass through that mesh. Some of your fibers or other really small particles might be able to wiggle their way through the holes in the mesh. So it's better at sampling the larger particles, things like fragments and spheres and films and things of that nature. We also did some simple grab samples, as you can see in this bottom left corner. We basically just took a amber glass jar and put it into a bridge sampler, a weighted bridge sampler that we used, tied it to a rope, lowered it off a bridge down into the water, directly filled that plastic jar with river water, capped it off so no contamination from our hands or clothing could get in there, and took that sample directly to the lab for analysis. And the advantage of these samples is that you're getting a whole water sample. Any plastic particle that's in that water is staying in there until you get it to the lab to analyze it. The big downside of the grab sample method is just the volume of water. You just can't sample the same volume of water that you can with the net sampler. We did, I think, 10-liter samples with the grab sample versus the thousands of liters that we were sampling with the net sampler. Finally, we have a Niskin sampler in the bottom right. This is a version of a grab sampler that we used that allows us to sample different levels of the water column. Basically, the sampler is lowered into the water on a rope. There's a trigger mechanism that holds the sampler open, and you send a weight down the rope that hits the trigger, closes the sampler at whatever depth in the water column you're looking to take a sample. And this allows you to take vertically stratified samples. So for instance, for this study, we were sampling the main stem Delaware down where it's 45, 50 feet deep in the navigation channels in the urban portion of the estuary. And we were able to take a sample at the surface and the bottom to see if there was differences in plastic concentrations at those two areas. So those are kind of methods on how you go out and you get your sample, but then what do you do when you get the sample back to the lab? So when you collect your sample, there's water in it, there's sediment, there's organic matter, there's a lot of things other than plastic in the sample. And so the first thing we do is we filter the water out from the solids, and we use a set of soil sieves to do that. This is nice because we put the sample through there, and then each layer of these sieves is a different mesh size, and so it kind of sorts your sample out by size as you're going through, in addition to separating the water from the solids. For a lot of our samples down in the estuary, there's a lot of suspended sediment, so we have a lot of sediment in the samples. And in order to remove that, we use what's called density separation, where basically you're taking all the solids in the sample, putting it into a salty water mixture, and all the plastics end up floating to the surface of that mixture, and all the sediment settles out at the bottom, and then you can either pour off the bottom or scoop off the top to isolate just the plastic particles. Additionally, we also got a lot of organic matter in the samples, especially sampling in the fall when leaves are coming down. With the net sampler, you're getting a lot of leaves in the samples. And in order to remove that organic matter, we used an oxidation process, basically pouring hydrogen peroxide onto the sample to break down that organic matter. But the hydrogen peroxide doesn't react with the plastic, so that's preserved for analysis. And so that's how you kind of go from having this sample that's got all sorts of river debris and everything else in it into just isolating the plastics. So once you get those plastics isolated, how do we actually study them? You know, what do we do in the lab? So you can do lots of things, everything from just a straight count. You can count by color or shape or size. And so the analysis is pretty much as simple or as complex as you want it to be. On the simple end of things, you can just use a microscope, look at these particles under a microscope. It sounds very simple, but oftentimes, you know, you look at this little thing that's the size of a period, you have no idea what it is. You put it under a microscope and it's obvious that it's plastic. You can also do a hot needle test where if you heat up a needle and touch the particle, if the particle melts or deforms, it's probably plastic. And if it doesn't, then it's most likely, you know, a piece of sediment or maybe a piece of wood or something like that. So those are kind of the simple ways that you can look at microplastics in the lab. You can also do more complex things like FTIR and Raman spectroscopy. And that's what we did in our study here. We used FTIR spectroscopy, which is, honestly, it's more complex than I know all the details of. But basically, you're scanning each individual particle with this very high-tech microscope. And the microscope calculates the spectrum that you see in this graph here. You see these two lines. The top line is the spectrum of one of our particles that came out of Assunpink Creek. The bottom line is the known spectrum for polyethylene. And as you can see, when you look at the peaks and valleys, they all match up really nicely, which suggests that the particle that we found was in fact polyethylene. And so for our data, we were able to go a step past just a count and a size and a shape and actually get the chemical composition breakdown of what the plastics we were finding, whether they're polyethylene, polypropylene, styrofoam, PVC, etc. So that's some basics on, you know, overall microplastics work and, you know, how you can go about doing a study. Now I'm going to give us a little more info on our specific study that we've done over the past couple of years. This is work that was funded by the NIFWIF Delaware Watershed Conservation Fund. And we set out to collect surface water samples in the Delaware estuary. We had 4 sites in the main stem river that were sampled by boat. And these are the sites where we used that Niskin sampler and took a surface and a bottom sample. We also had 9 sites that were in tributaries to the estuary, and 1 site in the non-tidal river in the Calhoun Street Bridge in Trenton to get an idea of plastics coming in from the non-tidal river. We used a combination of nets, grab samples, and Niskin samples. So at the outset to the study, we were focused on mainly grab samples. We were going to do those bridge grab samplers from the tributaries and the Niskin sample on the main stem river boat sites. About halfway through the study, we decided to add in the net samplers as well to be able to compare the results between the 2 methodologies. And we added those in at several of the tributary locations where we had conditions that were you know, favorable for that type of sampling. So wadeable tributaries that didn't have a lot of tidal influence. A lot of our tributaries in this stretch of the river are tidally influenced. So when you're deploying a net for, you know, 6 hours, we wanted to make sure that it wasn't going to end up high and dry after the end of a tide cycle. So those are how we collected the samples. And then the samples went to a lab for analysis, and we used the wet center at Temple University. They had done some microplastics work in the past. So they analyzed our samples for us using the FTIR spectroscopy that I had mentioned earlier, and that's the method where they will be able to produce an estimate of what that particle you know, actually is, whether it's polyethylene, polypropylene, or whatnot. And so the ultimate goal of the study is that we would collect this data and it would inform models that would allow us to identify high plastic loading tributaries, which would then be targeted for cleanup efforts with some of our partner organizations. Now, I will mention here before going any further that we did hit several hiccups throughout the study, mainly driven by shutdowns related to the COVID-19 pandemic. Our analytical lab was shut down for most of 2020. They were not in the lab analyzing the samples. So things did not quite go in the order we had originally planned in terms of data modeling, targeting cleanups. We ended up having to schedule the cleanups before we got the data back just to fit everything in the right time frame. But ultimately, we got everything done that we wanted to. And in the next slides, I will talk a little bit about some preliminary results. I will keep it brief. One, because I don't want to bog things down too much, but two, because we are still in the process of finalizing everything. So I don't want to share too much if we end up having to go back and change some things. So this first slide here is looking at plastic concentrations in particles per cubic foot at all of our sites with two different collection methods. So if we look at these graphs here, the top panel are all of our grab sampling sites, and the bottom panel are the paired net sample sites. And the y-axis is the number of microplastics per cubic foot of water, and the x is each one of the sites. So grab sample wise, we had the highest number of plastics in Rancocas Creek, up to about 250 microplastics per cubic foot of water. And that ranged all the way down to about 10 microplastics per cubic foot of water. And in the net samples, I will say important to point out that the concentrations we saw with the net sampler were drastically lower than the grab sampling method, orders of magnitude lower. And we are still kind of trying to disentangle why that is and trying to figure that out. I do think it has something to do with the fact that that net sampler does allow some of those smaller particles to pass through, and the grab sampler does not, and specifically the fibers. And so I'm going to show our next slide, which will be a breakdown of the types of the shapes that we saw, and it's going to kind of point to those same results there. So we've got another big confusing slide here, but I'll try my best to break it down. So we've got these colored bars here, and each bar represents a sample, and each color represents a different shape of plastic found in that sample. The top chunk here, the top 3/4 of this are all the grab samples, and the bottom quarter down here are the net samples. And as you can see, these grab samples are largely green in color, which relates to fibers. So we saw 95% or so of the particles that were found in the grab samples were fibers. At the bottom here are the net samples, and you see a much more diverse array of particles. You see a lot more fragments, a lot more films in the net sampler, and that could be driven by the fact that that net sampler isn't catching all the particles that are actually coming through there, because they're wiggling their way through the holes in the net. So as I mentioned, these are just preliminary results. We're going to have a lot more to share in the coming months as we finalize our report. Another portion of this study is going to be a web-based data viewer that we're developing. This here is a screenshot from that. I was going to link the real thing in here, but I didn't want to risk technologically, having to go to a web browser. So I just took a screenshot and, oops, sorry. Go back to that. And so I just put a screenshot in here for the sake of efficiency. But what we'll have here is will be a map-based web viewer that will allow you to see all the points that we sampled at. They are sized based off the plastic concentration, sized and colored based off the plastic concentration. You can also click on an individual point and you'll be able to see what the composition of that sample was. So for example, here we have a Christina River crab sample. It's probably very difficult to see the text on the screen, but we've got mostly fibers that were clear in that sample. And this also allows you to toggle between the grab samples and net samples to compare the two. And so that's something that'll be coming out when we finalize the report here in the next couple months. Okay, and as I mentioned, another portion of this study was these targeted cleanup efforts at high plastic loading tributaries. And so, as I mentioned, we kind of ran into some timing constraints due to COVID, and we had to schedule these before we had the the analytical data back. And so we just kind of went based off of our on the ground in the field knowledge of what we saw when we were out collecting the samples. And we picked a couple locations where we knew there was a lot of plastic debris along the shorelines, one of which was the Frankfurt Creek, which is, you know, as urban as it gets in Philadelphia. A large portion of the creek is actually concrete underneath it. It used to be basically a storm drain. So we did one cleanup effort there. And we also did one along the main stem Delaware River at Palmyra Cove, again, in that urban stretch of the river. And both of these efforts were very successful. We filled lots and lots and lots of bags of trash and picked up lots of plastic off the shorelines. We also were able to get out and talk to the volunteers that came out to help us with these cleanup efforts. And we did a little, you know, education on the topic of microplastics in our efforts to study them. So that's about it for our study. I will just briefly touch on a couple other studies in the basin on plastics, specifically this USGS study, microplastics in the Delaware River. This was a study I believe they completed last year, and this focused on the non-tidal river. So they took samples from, I believe, Washington Crossing all the way up to Narrowsburg. I think they had a site at Port Jervis as well. And so they're more up in your guys' neck of the woods with their study. And they looked at plastics in water, sediment, fish, and I believe also in mussels. And so that was a great study that came out last year. And there's also a study by the University of Delaware, John Cohen, his lab at the University of Delaware, where they looked at microplastics down in Delaware Bay. So between these two studies and our study, we kind of have a good coverage of the river now in terms of some preliminary looks at microplastics in the whole basin. So there's a lot of talk there about, you know, numbers and plastics and methods and all that type of stuff. But so like, how can we help and why should we help? So why? There's just some, you know, not so fun facts about plastics. 91% of plastic ever made was not recycled worldwide. And since 2012, we as the United States are only recycling about 9%, which is less than some of our counterparts overseas. One prediction that's out there is that by 2050, there's going to be more plastic in the ocean, ton for ton, than fish. And plastic can take more than 400 years to decompose. So it's important to, you know, get it out of these systems because it's not going anywhere on its own. And just some suggestions on how we can help conserve water and reduce plastic waste. Things like using refillable coffee mugs, refillable water bottles, reusable bags, saying no to straws or bringing your own reusable straw. They now have BYO, bring your own carryout containers, and also participating in volunteer cleanup efforts in your local watersheds, similar to how we did here in this study. And that's all I've got for tonight. Feel free to let me know if you have any questions, and if you can't think of any now, but you remember them later, feel free to reach out to me at my contact on the screen there.
Looks like Ginny, did you have a question?
Yes, Jake, I know you mentioned there has been a study done in the Port Jervis area. I was just wondering if you have any information on how the upper Delaware may compare to the lower watersheds as far as microplastics.
Sure. So they definitely found plastics in the upper Delaware. I believe I would have to go back and check the numbers, but the concentrations were on par with what we were seeing in the lower main stem. You know, I may be a little lower in the upper Delaware, but they were found, I believe, at all sites in the upper Delaware. It gets a little tricky because their methods were a little different than our methods, so it's hard to compare them directly, but they did find plastics, I believe, in all water, sediment, and fish sampled in the upper Delaware.
Thank you.
No problem.
Ashley, comment? Can you all hear me?
Yep.
Okay. I just wanted to, I thank you for your presentation. And in town of Tustin, we've been recycling plastic through a program that Trex, the people that make deck plastic, you know, make decks and so forth, offer. And two things, as a two-person household, it's incredible how much plastic I recycle through the Trex program, bags and bags full of it. And it's a 2-person household. So there's a lot of plastic in our lives. And then the other good part is the town has 2 benches or 3 benches on the deck. They're green, they're very attractive. And we have a couple of benches in the gazebo. So it's really, I think it's a good program. And I can connect you with our energy committee person who started it. So I just wanted to put that out there.
Thank you, Susan.
I have a question. Hello?
Go ahead.
Yeah, I know, like with the Hudson, the New York State held GE responsible for the PCPs they had been dumping in over the years. Now I know the lower Delaware and some of the tributaries, they have Haas and Roman Haas was down there, and I know DuPont Chemical and a lot of other these heavy manufacturers. Has any thought been given as to going after those people to help clean up?
And there has been on some of those other topics like PCBs. On plastics, though, I think it's just hard to trace that source if it's coming off of city streets and getting washed into the streams. I mean, you guess you could go after the city of Philadelphia and say you need to clean up the trash better, but that's a big ask.
Brad, this is Joe. I will tell you that the Park Service does have a contaminated sites division out of Washington, D.C. Now, again, it's only pertaining to Park Service property or anything that would affect Park Service property. But I know that they've had litigation against different companies over the Potomac and the dumping that's been going on in the Potomac. And they've had a multi-million-dollar settlement over that. So the Park Service does have a contaminated sites program that looks into a lot of this. I mean, we could talk more later, Fred, if you want to.
Yeah, that'd be good. Because I have a heavy background in industrial plastics. And I know a lot of these we dealt with. And I know that before the EPA, without filtering or without any thought of what they were doing.
But like
That's
Jake
why I was
said,
just
it'd be tough to
curious.
track it back to a specific company with the plastics. A lot of the stuff that they're pulling out of the Potomac is pretty obvious on, you know, who did it. It was pretty easy to track down. So.
Well, thank you.
Any other comments or questions? I have one for Jake, and that is, do you know of any funding for trying to get the plastics out before they get in the river, to get out of the environment, cleanup programs, or any funding or help on cleanup programs?
Off the top of my head, no, but I can look into that and let you guys know if I could find anything. I'm sure there's something out there.
The second question I have is, we're talking about measuring and trying to get a handle on the quantity and whether it's more or less in the worst spots. But what about the medical aspects of it in the food chain? Do you have information regarding, you know, I read in certain articles that basically microplastics are on almost throughout the food chain, and even in human livers, et cetera. If you go to the vortex in the oceans and go 5,000 feet down to the bottom feeders, they have microplastics in their livers and things like that. Do you have any information on the deleterious effects of plastics in the food chain and on the human species?
Yeah, so I don't have anything off the top of my head. It is very much an emerging field of study, and there are studies coming out. I know in the beginning, a lot of the studies were kind of saying that the plastics could be inert and they would just pass through your system. But I think now they're starting to see more often that, you know, they are accumulating and that there could be some negative effects. Although, you know, I don't have a study to give you off the top of my head, but I can look into that as well.
If there are no other questions for Jake, we want to thank you for your presentation. And by the way, so you're building this data. What is the plan? How long does this study go on? You know,
So the study is over right now. We are just wrapping up the final report for it. And so in the next couple months, it'll all be wrapped up and available to the public. And there could be future studies that come off of this, but what was the work that was funded a couple years ago is wrapping up right now.
because I asked the obvious question, wouldn't you wanted to know if the plastics are diminishing or increasing in the Delaware River system?
Yeah, it definitely could be a topic of long-term monitoring, and just from working down here in the urban portion of the estuary, the amount that we removed on our cleanup efforts is just a drop in the bucket on the overall issue.
And thank you very much for your presentation.
Oh, no problem. Thanks for having me.
Okay, let's move on with our agenda.